Measuring the brain secretions. In order to determine what NT or neuromodulators are being released in the areas of brain, the microdialysis is one of the methods that analyzes the chemical present in interstitial fluid through a small piece of tubing made of semi-permeable membrane that is implemented in the brain. The brain, in addition to its electrical activity, also produces and releases variety of chemical substances known as neurotransmitters and neuromodulators. And these substances play critical roles in communication between neurons and in regulating various brain functions. Measuring the brain secretion is essential for understanding the underlying mechanisms of behavior, cognition, and emotion. Direct measurement techniques involve microdialysis. This technique involves the implanting a probe into a specific brain region. The probe contains a semi-permeable membrane that allows small molecules such as neurotransmitters to diffuse into probe. A perfusate solution is continuously pumped through the probe and the concentration of neurotransmitter in the perfusate can be measured using various analytical techniques such as high performance liquid chromatography, HPLC, or mass spectrometry. In vivo voltammetry, this technique involves implanting microelectrode into specific brain region. The electrode is made of material that can oxidize or reduce neurotransmitters. By applying a voltage to electrode, researchers can measure the current produced by the oxidation or reduction of neurotransmitters, which is proportional to their concentration. The indirect measurement techniques involve immunohistochemistry. This technique involves labeling neurons on their processes with antibodies that bind to specific neurotransmitters or their receptors. By visualizing the location of these proteins, researchers can infer the presence and distribution of neurotransmitters in the brain. Radioimmunoassay RIA. This technique involves using radioactive isotopes to label the neurotransmitters or their antibodies. By measuring the binding of labeled neurotransmitters or antibodies to their targets, the researchers can quantify their levels in biological samples. Enzyme linked immunosorbent assay ELISA. This technique is similar to ERIA but uses enzymes instead of radioactive isotopes to label neurotransmitters or their antibodies. The neuroimaging techniques such as positron emission tomography PET and single photon emission computed tomography SPECT can be used to measure the level of neurotransmitters in the brain by using the radio labeled ligands that bind by using the radio labeled ligands to specifically to the specific neurotransmitter receptors. Challenges and limitations. Measuring brain secretions is a complex task with several challenges. Invasiveness. Many techniques such as microdialysis and in vivo voltammetry require the implantation of probes into the brain, which can be invasive and cause damage. Sensitivity. Some techniques may have limitations in terms of sensitivity, making it difficult to detect high levels of neurotransmitters. Specificity, it can be challenging to ensure that the measurements are specific to the target neurotransmitters and not affected by other substances. Dynamics, the neurotransmitter level can fluctuate rapidly in response to various factors, making it difficult to capture their dynamics. Despite these challenges, the development of new techniques and advancements in analytic methods are enabling researchers to make significant progress in measuring brain secretions. These methods are essential for understanding the underlying mechanisms of brain function and for developing treatments for neurological and psychiatric disorders. So next is the stimulating neural activity. The stimulation activates the neurons by electrical or chemical stimulations through a cannula, small metal tube that can be inserted into the brain of a lab animal. Stimulating neural activity helps in modifying the behaviors and also for the cognitive processes.